Everyone, welcome back to our locomotion and movement chapter. So we'll be carrying forward with the concepts of movement and locomotion from where we left in our previous video. Just to give you a quick recall of what we understood in the introduction of this chapter, I told you that a muscle would be basically consisting of what? It would be a collection of structures called the fasciculi. So here is, let's say, an entire muscle. So what exactly is the muscle made up of? Many bundles like this. So these are the bundles inside the muscle which are called the fasciculi. So every single structure here is a fasciculus. And if we can recall, what were the fasciculi themselves made up of? They were collections of individual cells called the muscle fibers. So here is basically one individual cell, that is your second individual cell, and here's the third cell. Okay, so each cell here is basically called a muscle fiber. So why should we call a cell as a fiber here? Because the cell is elongated in shape. Okay, just to highlight them in different colors so that you can actually identify them. This is your one cell, here is the second cell, that's your third cell. So these are three individual cells or the fibers, all of them inside a single bundle called the fasciculus. On the lower side also, here's your cell number 1, cell number 2, and cell number 3. And one more thing that we can recall over here is how are the individual cells packed together. What is this connective tissue that is holding the cells together? That is endomysium. Here also, what is holding the individual cells together? What is acting as the packing material between the cells? That is your connective tissue called the endomysium. Okay, then if you talk about the second layer of the connective tissue, the second layer of connective tissue would be holding the fasciculi together. This one, there would be a connective tissue here also. This is holding the fasciculi together. There is a connective tissue here also surrounding the fasciculus, and there's a connective tissue here also surrounding the fasciculus. So this connective tissue that is surrounding your fasciculi, or that is packing the fasciculi together, this was your perimysium. In this gap also I would be having the same endomysium, because the connective tissue inside a fasciculus, or the connective tissue holding the various fibers together, filling up the gaps between the fibers, that is all endomysium. And the connective tissue holding the fasciculi together would be perimysium. And there is also one more connective tissue which should be covering your entire muscle overall. This one. This connective tissue is covering your entire muscle. So the connective tissue covering of the entire muscle, what is that called? Epimysium. So this is what we know till now, that a muscle is a collection of fasciculi, and each fasciculus is a collection of cells called the muscle fibers. How are the cells held together? By endomysium. How are the bundles of the cells held together? By perimysium. And how is the entire muscle covered? By epimysium. Okay, so going forward from over here, we also remember that since the name of the chapter was movement and locomotion, if this is, let's say, somebody who has to actually move from one place to another, what do you require for locomotion? Movement of your legs, of course. So how do your legs move? With the help of the muscles present in them. And what type of muscles do we have in our legs? Striated muscles. So that's why we are basically going to focus on the structure of the striated muscle today, because we want to understand the mechanism of locomotion. To understand the mechanism of locomotion, we must understand how our striated muscles contract. But to be able to understand how our striated muscles contract, we should be first understanding the structure of the cells of the striated muscles called the muscle fibers. So in this picture, if I want to mark a single individual muscle fiber, here it is. This is your single muscle fiber of a striated muscle. So if I look at the properties of a single individual muscle fiber of a striated muscle, I can see that it is elongated in shape, cylindrical, as you can see from the shape, plus there's one more important property about the striated muscle fiber. It is syncytial, which effectively means multinucleated. Now I would give you a background story about what exactly is the meaning of the word syncytial, because you know that this channel is not exactly to just give you the facts, is to basically help you understand the concept behind each and every word. Okay, so what exactly is the meaning of word syn? Fusion in biology, and cyto, of course, are what? Cells. So if I go back to embryonic stages of humans, in the human embryo, we have got certain cells called myoblast cells. Each myoblast cell is basically a small normal cell with a nucleus, one more myoblast with a nucleus, one more myoblast with a nucleus. These are also called the precursors of the muscle fibers. Myoblast cells are the precursors of the muscle fibers. If you can recall, there's a similar term that you have studied in connective tissue, osteoblast, the cells in the bones that give rise to new bone material. Similarly, chondroblast, the cells that give rise to new cartilage material. So here's myoblast, the cells which are precursors of the future muscle fibers. So what happens in the embryonic stages is these precursors fuse together. So now you see cells fusing together, syncytium. Okay, as the cells fuse together, what do they give rise to? They form a multinucleated cell. To form a multinucleated cell called the muscle fiber. So when I fuse this and this cell together, what do I get? This kind of a cell with how many nuclei in it? 3, and of course their common cytoplasm. This is your muscle fiber. Here, elongated, cylindrical, syncytial. So if I want to show the nuclei here, this is how I can actually show the nuclei. Is it compulsory that a muscle fiber will be formed by fusion of 3 myoblasts only? No, that is just a random number I've drawn. There can be 4, 5, 6, all of them fused together. So then the muscle fiber will be having 4, 5, 6 or 7 or whatever number of nuclei. So this is just a random number that I've chosen. Do not think that a muscle fiber will be made up of 3 myoblasts only. No, it can be any number, and that basically is going to determine the size of your muscle fiber. So here is your striated muscle, and these are the units of the striated muscle called the fasciculi, and inside each fasciculus there are the individual cells called the muscle fibers that look elongated, cylindrical, syncytial. Okay, now if we look at the muscle fiber, the cell, in further detail, we would be able to find out a few more interesting facts in it. What are those? Let's see. The elongated cylindrical syncytial cells called the muscle fibers, they would also be having cytoplasm in them, just like any other cell. The muscle fiber obviously will be having cytoplasm, but the cytoplasm in case of the muscle fiber is called sarcoplasm. Plus their cell membrane, what is that called? The cell membrane is called Sarcolemma. For a normal cell, what is the cell membrane called? Plasmalemma. For them, what is the muscle cell membrane, a muscle fiber cell membrane called? Sarcolemma. So the same cell, which is this one over here, if I actually draw it in a magnified view in the sense, I would basically draw it like this. Okay, here I am basically doing what? I'm just drawing a part of the cell. Let's say this much part of the cell I'm drawing over here in detail. That is why I've kept it open on both the sides, because we're just drawing a portion of the cell, not exactly the entire cell. Okay, so in this what we can show is the labelings of various parts. The cell membrane here, what should we call it? Sarcolemma, as I told you. Then there'll be of course a few nuclei in this portion. Here are the nuclei. A single one is nucleus, the plural for that is nuclei. There is of course the cytoplasm also. And what exactly is this cytoplasm called? Sarcoplasm, right? All right. Now, one more thing that we would be able to see in this magnified structure is something that actually helps us to explain why they are called striated muscle fibers. What is the meaning of the striations? All of you have seen the zebras, right? The animals. So if you talk about a zebra, what do they have? Alternate dark and light stripes on their body. Similarly, if you have seen the roads, there are zebra crossings on the road. So what is basically present on a zebra crossing where people cross? Alternate dark and light stripes. So there are similar dark and light stripes, also called striations, which are visible on this cell also. Because of these visible dark and light stripes or striations, these are called the striated or the striped muscles. So when I talk about the striations, how exactly will they be visible on the surface of the muscle fiber? Something like this. So as I see, repeating at regular intervals on the surface of the muscle fiber are structures which are called the striations. So it's these stripes or the dark lines with some light areas also between them. That's the reason. This kind of appearance is the reason why these muscle fibers are called striated muscles. Now you might be thinking, what exactly is the reason behind these? That's what we are going to cover in this lecture. Okay, so on the surface of the striated muscle, I can actually notice that there are alternate dark and light stripes. Stripes, also called striations, which is the reason why they are called striated muscles or striped muscles, or in this case this is not exactly a muscle, this is a muscle fiber. But then yes, if fiber is like this, the bundles of such fibers are called fasciculi, and one more fiber, the bundle is called fasciculi, and the bundle of bundles, that is the bundle of fasciculi or a group of fasciculi, that would be your entire muscle. Okay, so if the fiber is like this, obviously the entire muscle also will be having what? Similar stripes visible on it. Okay, so these are the alternate dark and light stripes or the striations which are responsible for the name, the striated muscle fibers. Now let's move on to the fact that why will there be these striations over here? There has to be some reason, obviously, right? That there are some areas which appear like dark lines, and then there are some areas which are appearing much more lighter in density or in color. So why is that so? To understand that, we have to basically delve deeper or go deeper inside this cell. Inside this cell means where? Inside the cytoplasm of the cell here. You know from biomolecules that what are the building blocks of the cells? Proteins. So what do you think will be present inside the sarcoplasm over here? Proteins, of course. Okay, so if this structure over here that I've drawn is a fiber or a cell, or in fact we can say a part of the fiber or the cell, what should I call the proteins as? If the cell is a fiber, the proteins that are present inside the cell should be called fibrils, the subunits or the building blocks of the fiber. So let's move on to the reason why there are these alternate dark and light stripes. Inside the cytoplasm of this muscle fiber, what do we have? Elongated protein rods or we can say proteins arranged in the form of rod-like fashion. Let's say there's a protein like this here. There's a protein, I mean not a single protein, obviously collections of many proteins, because a single protein can't be that big. Collections of proteins or bundles of proteins. This is one bundle of protein, this is the second bundle of protein. So various protein bundles are present inside the cell. These protein bundles or rods are called myofibrils. Units of the fiber, fibrils or fibrils, whatever you want to pronounce it as. Elongated protein rods or protein bundles called myofibrils are present. Okay, and the reality is that the alternate dark and light bands that we have drawn on the surface of the cell over here, these bands, they are in fact present on the proteins which are present inside the cell. And the sarcolemma is somewhat kind of, let's say, transparent. So if there are proteins inside the cell which have got stripes on them and the cell membrane is transparent, will we be able to see those stripes through the transparent cell membrane? Yes, we can. Okay, so the reality is that all these stripes that we can see over here are not actually present on the cell. They are in fact present on what? The proteins inside the cell. Let's see how. The alternate dark and light bands are actually present on these myofibrils. So I hope you are not getting confused here. Fiber is the cell. The proteins are fibrils. And the stripes that we can see on the cell are actually present on what? The proteins inside. So how do we see them on the cell? Because imagine the cell membrane is transparent, glass-like. So whatever proteins are there inside, you can see those proteins through the cell membrane. So let me draw it again. Here's your sarcolemma of the muscle fiber. Okay, and what I've just told you is that there are elongated protein rods which will be present inside this cell like this. Here, this is one protein rod that is present in the cell. Then here's the second protein rod present inside the cell. Here's your third protein bundle or rod present inside the cell. Of course, the rest of the stuff will be still visible. Will you be able to see the nuclei? Yes, of course, close to the membrane, as we've drawn in the upper diagram. Will there be cytoplasm also? Of course, yes, it will be there. So as we have zoomed in inside the muscle fiber now, we can see that the muscle fiber or the cell, which is this entire structure, this is your fiber or the cell. So what does the fiber or the cell contain? These structures. What are these? What is this one single structure? A myofibril. What is this also? Another myofibril. What is this as well? Another myofibril. So these are the myofibrils. And as I was telling you a while ago, the alternate dark and light stripes that we saw in this picture, these stripes are actually present where? On these proteins, on exact same locations. Okay, so if I want to actually represent them on the places where they actually are, or on the structures that they actually are, it would be something like this. I would just draw the markings first here. Now let me zoom in over here. So what do you think is this? These are the stripes, the alternate dark and light stripes. here also on this one as well. And on the last one also. So can you see that where exactly are the dark and light lines present? On the myofibrils. So if I zoom out like this, where do you think the dark and light stripes actually are located? On the protein rods. But then if we are actually looking at them through a transparent cell membrane, you would be actually let's say believing that the entire cell has got what? Stripes on it, while the reality is the stripes are exactly not on the cells. Where are they? We can see on the protein rods or the myofibrils, right? So in this picture what I can see is a cell with dark and light stripes on it, but when I zoom in I find out that these stripes are not exactly on the cell. They were just visible on the cell because the cell membrane was somewhat transparent, but in reality where exactly are these dark and light stripes located? They are located on what? On the myofibrils, not exactly on the muscle fiber. But then you got my point, right? Because the cell membrane is somewhat transparent, the dark and light bands seem to be present on the surface of the cell itself. Here's your cell membrane. Let us make it a bit thicker to be able to understand that this is the boundary of the cell. So here's your cell. These are the myofibrils inside the cell. Where exactly are the dark and light stripes? On the myofibrils. But when you look at it in a zoomed out picture, now it looks like that if this is your entire cell, the stripes seem to be present on the cell itself as drawn in this picture over here. But in real view, when we see, we find out that the stripes are actually on the fibrils and they are visible through the cell membrane. Okay, so this picture tells us that the myofibrils have got the alternate dark and light bands on them. Now we need to understand that why will the fibrils have the alternate dark and light bands? The fibrils, that is the proteins, have got the alternate dark and light bands. Why so? To understand that, let's go deeper. Let's take one single individual muscle fiber, any which one of your choice. Let's take the green one. If we talk about an individual myofibril that contains two types of subunits called thick or primary myofilaments and thin or secondary myofilaments. An individual myofibril itself contains two types of subunits. What exactly is a myofibril? A collection of proteins. So what will be its subunits? Proteins only. So there are proteins arranged inside a myofibril in two manners: thick or primary myofilaments, thin or secondary myofilament. One problem over here is NCERT has mentioned that inside the cytoplasm of a muscle fiber, elongated protein rods or bundles called myofibrils or myofilaments are present. That is what NCERT has mentioned. So according to the NCERT, myofibril and myofilament is the same thing. However, the reality is the muscle fibers are the cells. The proteins inside the cells are called fibrils, and the fibrils have two types of subunits in them: thick and thin filaments. But then anyways, since fibrils are made up of filaments, fibril and filament has been used as alternate terms in NCERT, and you can always choose that as an option if you get a question where no other option is correct. So let me tell you something about this myofibril over here now. An individual myofibril I've mentioned contains two types of subunits called thick or primary myofilaments and thin or secondary myofilaments, and a particular arrangement of these myofilaments, a specific particular arrangement of these myofilaments is actually responsible for the alternate dark and light bands. So this is the place where we will actually understand the reason why dark and light bands are there on the myofibrils, which are visible on the entire cell or the muscle fiber also. A myofibril contains two types of subunits called the thick and the thin myofilaments, and a particular arrangement of these myofilaments is responsible for the alternate dark and light bands. Let me tell you how. As I was telling you, let me choose any protein or any myofibril. Let me take the green one here. Okay, so if I take the green myofibril and draw it over here, that's your first myofibril, and whatever is applicable to one myofibril will be applicable to all of them. Once again, any numbers that I'm drawing over here, they're just randomly chosen numbers. Do not be under the impression that a muscle fiber will be just having 3 myofibrils. I'm just choosing any random numbers over here. A muscle fiber, depending on its thickness, may have 10, 15, 20 more or less number of myofibrils. So here is your one single myofibril, which, as you can notice, has got what? The dark and light bands on it. Okay, so what exactly is responsible for these dark and light bands? I'll tell you. So this myofibril is actually a collection of two different types of subunits called the thick and the thin myofilaments. Okay, so what exactly are the thick and the thin myofilaments? Let me show you. Now this would be actually going a bit deeper inside the structure of the myofibril. Okay, so here we go. I'll basically show you a zoomed in image also. Just keep observing what I'm doing as of now. And this is something that you won't exactly be able to find anywhere else. This kind of picture that I'm showing you. Why? Because it's important to be able to understand the structure yourself, then only you can explain it to others, which unfortunately, you know that a lot of people don't. Well, yeah, so here's basically your structure of the myofilaments of two types, the thick and the thin myofilaments. And in a while you'll be able to understand what exactly I'm doing over here and why is your myofibril showing that alternate dark and light appearance. So that's another thick or the primary myofilament. Here's one more thin myofilament. And that's another thick filament over here. Here. Okay, so let me first zoom in into this picture. What did I say? That an individual myofibril, the green thing was a myofibril. This was actually a collection of two types of subunits called thick or primary myofilaments and thin or secondary myofilaments. In this picture, let me actually label them for you now. The orange color structures that are drawn here are your thin or secondary myofilaments. All these structures are the thin or the secondary myofilaments. Everything that is in orange color is thin or secondary myofilament. All these. And the structures that you see in the center are a bit thicker. That's why, what are they called? Thick myofilaments, of course. What else? Here. These structures in the center are called the thick or the primary myofilaments. Okay. Now that's basically the arrangement of the subunits of an individual myofibril, thick and thin myofilaments. You'll be surprised to know that there is more detail. The secondary myofilaments consist of three types of proteins: actin, troponin, tropomyosin. The thick myofilaments contain only one type of protein, myosin. Those are further subunits of these myofilaments. So can you see the complexity here? A muscle is made up of fasciculi. Fasciculi are made up of fibers. Fibers have got what? Fibrils. Fibrils are made up of what? Filaments. Filaments have got what? Individual proteins: actin, myosin, troponin, tropomyosin. So we'll do all these details. We are at what level right now? Till over here, and we are going on to the next level, that is myofilaments. So what I've drawn in a myofibril, this entire structure here is a myofibril. And what is a myofibril made up of? Two types of subunits: thin or secondary myofilaments, thick or primary myofilaments. Now look at their arrangements. It's a fixed pattern that is repeating after every short interval. And now what I have to do is basically imagine this myofibril as maybe a pen. You put this pen on basically a source of light. I mean, you put the source of light behind this pen and observe it from the front. It's like basically hold a pen against the light actually, and you will be able to see that there will be certain parts of your pen that would actually appear lighter. There are certain parts of your pen that appear darker. Why? Because there are certain parts of your pen that actually are going to have more light passing through them. There are certain parts that will be having less light passing through them. If you're not able to understand what I'm saying over here, imagine this is your ballpoint pen with which you are writing here. Okay, this is the tip of that pen over here, right? And here's the refill which is running through this pen, and here's the pointed tip with which you are writing. So since the refill is filled with the ink, you are holding this pen against a tube light. So do you think this part will appear lighter and this part will appear lighter? Yes. I mean, imagine the pen is basically made up of those cello pens with the glass body and a refill inside them. So since it's a glass or a transparent plastic body, the transparent plastic parts are all appearing what? Lighter, because light is passing through them. But what is the refill looking like? Darker, because light cannot pass through it because there is ink filled inside the refill. So it's exactly the same. You observe your myofibril against a source of light, you'll be able to notice that the places where protein density is high, the light absorbance at those places will be high. The places where protein density is lesser, the light absorbance will be lesser. And the places where light absorbance is lesser, they'll appear lighter. Places where light absorbance is more, they'll appear darker. Now let's compare the two structures here. If we see this portion now, just observe it carefully. In this much portion of your myofibril, what exactly is the protein density? Very high. In this much portion of your myofibril, what exactly is the protein density? Very high, because both the proteins are there, thick filament as well as thin filament. In these portions, the thin filaments are over here on top, on bottom also, and there is the thick filament also in the center. So in this much portion here, both the proteins are overlapping each other, thick as well as thin. So since the proteins are overlapping each other, how much light absorbance will be there? Very high. So what will be the appearance of these structures when you're observing them in the microscope? Darker. Darker because the protein density is very high. And if you look at this portion again here, this portion again here, once again the proteins are overlapping each other. So what do you think will be the color? Dark. Go further. Here, here the proteins are overlapping each other. So what do you think they'll look like? Dark. Proteins overlapping each other. What does it look like? Dark. In the center of the two dark areas, there is only thick protein. In the center of the two dark areas, there is only thick protein, just one single type of protein. And in these areas also, there are only one type of proteins. What are those? Thin. So if there is only one type of protein here, thin, if there is only one type of protein here, thick, these areas are going to be basically absorbing lesser light and hence will appear what? Lighter. Okay. So now in the areas where the proteins, the secondary, that is thin, and primary, that is thick, myofilaments, the secondary and the primary myofilaments both are present. So these areas appear dark. But if you look at this area and if you look at these areas, only one type of proteins are present here. Only one type of myofilaments present here. So what do these areas look like? They are comparatively lighter. Areas where only one type of proteins are there, lighter. Areas where two types of proteins are there, darker. Now compare this picture with the picture on top. What was I telling you? This is one individual myofibril that I've taken. This is your myofibril that I've taken. Now look at this. There are certain portions in this myofibril where both the myofilaments are there. So they are looking darker. Both the myofilaments are there, darker. Both myofilaments are there, primary and secondary, or thick and thin, dark. Both myofilaments are there, dark. This area, over here, what is present? Only one type of protein, so they are appearing what? Lighter. Between the two dark bands also, there is an area with only thick filament, so that is also appearing somewhat what? Lighter. So if I mark the names here this much portion of your myofibrils is called the dark band. That's another dark band, another dark band, another dark band of the myofibril, and this much area from here till here is the light band of the myofibril, light band of the myofibril, light band of the myofibril. Once again, compare the two structures. Both the proteins are there. Even though there is a slight central area where only one type of protein is there, but this entire portion is dark band. This entire portion is called the dark band. This entire portion here is the next dark band. This is what? The next dark band. So why are they dark? Because we have got both the types of proteins overlapping each other in most of the areas in these bands. But here only thin myofilaments, light band. Here only thin myofilaments, light band. Here only thin myofilaments, light band. Okay, interesting. So I hope guys that you got this point here, that what exactly is a muscle made up of? Fasciculi. What are the fasciculi made up of? Fibers. What do the fibers contain? Fibrils. What do the fibrils contain? Two types of filaments, and those filaments are arranged in such a way that they will be responsible for the dark and light bands on the myofibrils. Each myofibril has got dark and light bands which will be visible on the entire muscle fiber and hence the entire muscle also, right? So I think that explains to you why exactly are the myofibrils striated. Now in our next lecture, what we would take up is basically the further detailed structure of the filaments. The thin or secondary myofilaments, as I've told you, are collections of three types of proteins. The thick or primary myofilament is a collection of a single type of protein molecule, myosin. So all this for the next lecture. Till then, keep studying and I'll see you later. Thank you for joining. Keep spreading the love, keep subscribing to the channel, share it with your friends, and keep watching for more interesting deep knowledge. Thank you. Bye bye.